Welcome to our project on serotonin. Serotonin is a monoamine neurotransmitter, which means that its structure consists of one amino group connected to a ring by a two-carbon chain. It is primarily found in the gastrointestinal tract, blood platelets, and the central nervous system of animals. In the enterochromaffin cells of the gastrointestinal tract, where approximately 90% of the human's body's serotonin is located, serotonin is used to regulate intestinal movements. In the central nervous system, serotonin is synthesized by serotonergic neurons, which are located in the raphe nuclei of the midbrain, pons, and medulla. In the central nervous system, serotonin is involved in the regulation of mood, happiness, appetite, and sleep. It also plays a role in cognitive functions such as memory and learning. The synthesis of serotonin begins when tryptophan is taken up by serotonergic neurons. In these neurons, an enzyme called tryptophan hydroxylase adds a hydroxyl group to a tryptophan, producing 5-HTP, or hydroxytryptophan. This structure is then further decarboxylated to produce serotonin, or 5-HT here. The serotonin is then packaged into vesicles and released into the synapse, where it binds to serotonin receptor molecule on the postsynaptic terminal. The SERT, or serotonin reuptake transporter, then reabsorbs serotonin into the presynaptic terminal to be recycled. SSRIs, or selective serotonin reuptake inhibitors, prevent the serotonin transporter from uptaking the serotonin prematurely or from taking the serotonin too quickly. Serotonin is a neurotransmitter that can have a postsynaptic effect if it just remains in the cleft, so it is important that serotonin is regulated correctly, both in its release and transportation back into the presynaptic terminal. Drugs that affect serotonin primarily target the SERT and SSRIs, but this will be discussed further in a later slide. There are 15 types of serotonin receptors, which can be grouped into three main families based on the way they operate: 5-HT1, 5-HT2, and 5-HT3, and are either excitatory or inhibitory. 5-HT1 receptors are inhibitory and have the greatest affinity for serotonin. When serotonin binds to these receptors, the receptor shape is altered, activating the G protein, which then opens an ion channel, allowing potassium ions to exit the postsynaptic neuron. This ultimately makes the threshold more difficult to reach. 5-H2 and 3 receptors are both excitatory. Serotonin binds to these receptors only after 5-HT1 receptors are saturated. In the 5-HT2 receptors, the activated G protein causes a potassium channel to close, resulting in the buildup of potassium ions inside the cell, depolarizing the cell membrane. Unlike 5-HT1 and 5-HT2, 5-HT3 receptors are comprised of binding sites and ion channels. When serotonin binds to these receptors, the central channel of the receptor opens, allowing sodium ions into the postsynaptic neuron and depolarizing the membrane. I just want to briefly mention that a study conducted at Yale explored the effects of serotonin on neuron firing in the frontal cortex of a rat brain. The recording showed a great increase in spontaneous excitatory postsynaptic currents, or EPSCs, in layer 5. This suggests that serotonin affects specifically the frontal cortex output neurons. Now I'm going to address some of the diseases associated with serotonin. Serotonin deficiency, which can be genetic or caused by lifestyle changes, causes depression, OCD, anxiety, and IBS. Because serotonin regulates mood and happiness, a depletion of serotonin most commonly causes depression and anxiety. One form of depression, called SAD, is related to changes in season. It's because sunlight affects the release of serotonin. Therefore, SAD usually begins around fall and ends around springtime. Because serotonin facilitates communication between the brain's deeper structures and the front part of the brain, a deficiency of serotonin, disrupting this communication, triggers OCD symptoms. Lastly, we want to mention some common drugs associated with serotonin. Depression is often treated with selective serotonin reuptake inhibiting drugs that increase the SSRI concentration in the brain. This effectively increases the amount of serotonin that contacts the serotonin receptor protein and prolongs serotonin's presence in the synapse. As a result, the effects of serotonin are magnified, thus stabilizing mood and happiness. Fluoxetine is a common drug prescribed that undergoes the process outlined above, shown in the diagram here. So we have serotonin going into the synapse and then properly being regulated by the reuptake transport protein here, so that not a lot of serotonin stays in the synapse. In this case, fluoxetine is bound here to the receptor, to the transporter protein, and the serotonin cannot be uptaken back into the presynaptic terminal, so a lot of it just stays in the cleft. Cannabinoids and opioids are among the drugs that affect serotonin levels. Cannabis, for example, increases serotonin at low dosages by inhibiting the serotonin transporter with SSRIs, as demonstrated here, similar to the fluoxetine. It should be noted, however, that at high doses, there's actually a decrease in serotonin levels, most likely due to alterations in cannabinoid receptors and in membrane microviscosity. We hope you enjoyed the presentation. Here's our bibliography.